In the monocots, the xylem will be more, wherein they'll have polyarch xylem condition, that is more than 6 xylem will be present. They have usually 4 xylem and therefore 4 phloem patches. So this is one of the characteristic feature of dicot root. In dicot root, it was inconspicuous, very small. But in monocot root, it is large and well developed. Innermost layer of the cortex of the root is the endodermis. So after the cortex, we have the layer, that is the roots have the layer which is called as the endodermis. Hello everyone, a warm welcome to the session on First PUC Biology. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, let us learn about the topic anatomy of dicot and monocot root, wherein we'll study about what exactly are the cells that are present inside the dicot and the monocot root, and later on we will move on to learning about some of the MCQs under this particular topic. So starting with the dicotyledonous root, so like how the outermost layer. So if you remember in the previous session, we had studied about in general what are the different layers, the cells that are arranged in a plant. So the cells are arranged in the epidermal tissue layer, then we have the ground tissue, and then comes the vascular system, right? So here, so therefore epidermal layer forms the outermost covering, and it is the epidermis. Epi means outer and dermis or derma means skin. So it is the outermost layer, and it is also called as epiblemma. And the cells of the epidermis or epiblemma, few of the cells, not all, they protrude or they modify themselves to form unicellular root hairs. So therefore, the root hairs that are there are unicellular. The trichomes can be multicellular. The root hairs, or what are trichomes? Trichomes are the hairs that are present on the, so here we are talking about the dicotyledonous root. So if you remember, in that is, you also have to know that in the stem of dicotyledonous plant, they do not have hairs, but instead it is called as trichome, or the leaf it is called as trichome, and they can be multicellular. But here in the case of root, in the case of dicotyledonous root, the root hairs are unicellular, wherein they are made up of a single cell. And next, below the epidermis, we have the cortex. So if you can see here, the outermost layer which forms the epidermis, and epidermis, it consists of parenchyma cells, but the shape of the parenchyma cells are barrel shaped. So it looks almost like this, like a barrel. And barrel is nothing but like in wine, that is preparation and all that, or beer preparation and all that. For fermentation process, they store it in huge drums. So that is nothing but the barrel. So it looks like that barrel. So that is why it is barrel shaped. So below the epidermis is the cortex. Now what is this cortex made up of? So the cortex that is there, entire this region is called as the cortex. So it comes between the epidermis and the endodermis or the pericycle. So what is the cortex made up of? So the cortex is made up of again parenchyma cells itself, but the parenchyma cells are covered by a thin layer. Therefore we can say it is a thin-walled parenchymatous cells and with lot of intercellular spaces. So they are made up of thin-walled parenchyma cells with lot of intercellular spaces like this. So this is how the cortex in the dicotyledonous root will be arranged. So next, moving on to the next part. So it is the endodermis. So after the cortex comes the endodermis. So this is the endodermal layer. Endodermis also, it is made up of parenchyma cells only. Here also the parenchyma cells are barrel shaped, just like how epidermis, the parenchyma cells were barrel shaped. Here also it is barrel shaped. And here it is made up of a single layer of barrel shaped parenchyma cells. The only thing is the parenchyma cells will not have any intercellular spaces. Even in the epidermis also, they are made up of a single layer of parenchymatous cells without any intercellular spaces. But here, why there are no intercellular spaces is because in the case of endodermis, because if we see the tangential and the radial wall, so here in the tangential as well as in the radial wall, that is the tangential wall as well as this is the radial wall, wherein we can see they are being deposited with suberin. There is a compound which is called as suberin that gets deposited in the tangential as well as in the radial wall of the endodermis, and that is why they do not have, or they're compactly placed. They do not have any space between them, between the parenchyma cells. So therefore, those parenchyma cells in the endodermis of dicotyledonous root having suberin deposition in the tangential as well as the radial wall, it is called as Casparian strip. So this is one of the important 1-mark question. And what is the function of this Casparian strip? So Casparian strip, they are made up of nothing but it is suberin deposition, and this suberin is quite waxy, therefore it acts as a water repellent which prevents the entry of water, or it is impermeable to water. So its main function is impermeability to water, or it acts as a water repellent. Next, talking about the pericycle. So pericycle are few layers of parenchymatous cells which are present below the endodermis. So you can see here pericycle, there are few layers of parenchymatous cells, again with intercellular spaces which are arranged just below the endodermis. So here they are made up of thick-walled parenchymatous cells and usually the lateral root. So if you remember, the root, it has the primary root. So this we call as the primary root. And then towards the sides, whatever root is being formed, we call it as the lateral roots or the secondary root. So primary root we call it as the mother root, lateral roots we call it as the secondary root. So these lateral roots, so the initiation or the development of the lateral roots as well as the vascular cambium during secondary growth takes place in these cells. So these are secondary growth, main root, then from there the secondary growths are the lateral roots. So these usually occurs from the modifications because of the cells, that parenchymatous cells that are present in the pericycle. So that is the function. And talking about the next layer, pith. So pith is usually the present at the center of the root or the stem. So here pith, it is inconspicuous. That is, it is quite small and it is inconspicuous. It is usually made up of parenchyma cells only with intercellular spaces. So next, talking about the vascular system. So the vascular system is nothing but it consists of the vascular bundles, that is the xylem and the phloem. The xylem which conducts water and the phloem which conducts the food. So here between the xylem and the phloem they have parenchymatous cells which are present and it is called as the conjunctive tissue. So this is one of the characteristic feature of dicotyledonous root which you don't find in the monocot root, that is the presence of conjunctive tissue. So what is this conjunctive tissue? It is nothing but the parenchyma cells present between xylem and phloem, we call it as the conjunctive tissue. So here if you can look at this diagram, we have the metaxylem, that is the xylem elements, and then comes the phloem. Between the xylem and the phloem we can find a layer of parenchyma cells or distribution of parenchyma cells are there which are called as the conjunctive tissue. So that is one of the characteristic feature of the dicotyledonous root. Next, usually dicotyledonous root will have 2 to 4 xylem and phloem patches. So if you can see here, they have, or we'll talk about the xylem. So they have how many xylem patches? So xylem patches is 1 here, 2, 3 and 4. So they have usually 4 xylem and therefore 4 phloem patches. So this is one of the characteristic feature of dicot root. How many xylem and phloem? 4 patches of xylem and phloem they will have. And later they will form a cambium ring between the xylem and the phloem. So between the xylem and the phloem, if a cambium ring forms, we call it as the open type vascular bundle, right? So they have a conjoint open type vascular bundle. So this is about the vascular system in dicotyledonous root. So next, moving further to monocotyledonous root. Monocotyledonous root also will have the epidermis, then comes the cortex, then comes the endodermis, the pericycle, the vascular bundles and then the pith. everything is almost same. So we'll discuss what are the differences that you get to find in the dicot route with when you compare, or difference between the dicot and the monocot. So there are differences in the vascular bundle. So if you remember previously I told you in dicot route they have 4 patches of xylem and 4 patches of phloem, right? So but in the monocot route they have more than 6, that is poly-R condition is seen here. Poly meaning many. So many vascular bundles or many patches of xylem and phloem are present. More than 6 patches of xylem and phloem are present. So if you can see here, 1, 2, 3, 4, 5, 6, 7 and 8. It is more than 6. So dicots will have only 4 xylem, 4 phloem, but in the monocots the xylem will be more, wherein they'll have poly-R xylem condition, that is more than 6 xylem will be present, xylem bundles will be present. So that is one of the main difference between the monocot and the dicot route. So here, but the pith in monocot route is large and well developed, but in dicot route it was inconspicuous, very small. But in monocot route it is large and well developed. If you can see here, the pith is large and quite well developed. So this is about the dicot and the monocot route. So next, moving on to some of the MCQs. So Casparian strips are found in. So where do we find it? We got to know that Casparian strips, they come under dicotyledonous route. So where are they present in the dicotyledonous route? They are present in the tangential and the radial walls of the endodermis, and therefore that endodermis we call it as the Casparian strip, right? So therefore it is found where? It is found in the endodermis of dicot route, not in the pericycle. It is the endodermis of the dicot route. Option C is the right answer here. Next question. The outermost layer of transverse section of root is cortex, epiblema, endodermis, pericycle. So it is epiblema, which is also called as epidermis. So epiblema is the outermost layer of the TS of root. So be it the dicot root or the monocot root, it is the epiblema. So that is why I have just given it as generally as root there. Next, the innermost layer of the cortex of root is, epidermis, endodermis, pericycle, stele. So innermost layer of the cortex of the root is the endodermis. So after the cortex we have the layer, that is the roots have the layer which is called as the endodermis. Next, Casparian strips are formed by dash deposition. Chitin, cellulose, suberin. Now, chitin you usually don't find it in plants. It is usually found in arthropods, wherein the exoskeleton is made up of chitin. Cellulose is of course found in plants. It is one of the main component of the plant cells, but they are not found, or they are not called as the Casparian strips. It is suberin, which are waxy. They are waxy components which are distributed in the tangential and radial walls of the endodermis, which we call it as the Casparian strip. So therefore Casparian strips are formed by suberin deposition. Option C. Next question. The anatomical layer in the root from which the lateral branches of the root arises is. So what is the question? The anatomical layer in the root from which the lateral branches of root arises is the. So we know that in the root there is the mother root or the primary root from which the lateral roots or the secondary roots develop. So from where exactly it develop? Does it develop from the endodermis, epidermis, pith or pericycle? So the lateral roots usually develop from the pericycle, wherein some of the cells, parenchymatous cells that are present in the pericycle, thick-walled parenchymatous cell, they undergo modification to develop into the lateral roots. So option D is the right answer here. Next question. The parenchymatous cells which lie between the xylem and phloem are called as. So usually we find it in dicot root. In dicot root, between the xylem and the phloem, they have few layers of parenchymatous cells. What is it called? Is it called as the vascular tissue? No, because vascular tissue is given the name given for xylem and phloem. Is it the conjunctive tissue? Yes, it is the conjunctive tissue. It's not the hypodermis, because hypodermis usually comes in the stem, and it's not the cortex as well. It's the conjunctive tissue. Option B is the right answer here. Next question. The types of vascular tissue system in dicot root is radial, conjoint, conjoint open, conjoint closed. So if you remember, I told you later on the vascular tissues in the dicot root, they actually between the xylem and the phloem, they form a cambium, right? So therefore it is a conjoint open types. Option C is the right answer here. So conjoint open type vascular bundle is present in the dicot root. Next question. All the tissues inner side to the endodermis is called. So all the tissues inner side to the endodermis is called pith, pericycle, vascular bundle, stele. So pith, pericycle, vascular bundle, all that comes within the endodermis itself. What is it collectively called? So collectively they are called as stele. So option D is the answer here. So next question. The vascular bundle in monocot root is it a diarch, triarch, tetrarch or polyarch? So we learned that tetrarch we find it in dicot root because they have 4 patches of xylem. But in the case of monocot root, the xylem numbers are more than 6. Therefore, they exhibit polyarch condition. So the right answer here is option D, polyarch. Next question. Which of these is not a characteristic feature of monocot root? What is the question? We have to find out of the given options here, which is not a characteristic feature of monocot root. 2 to 4 xylem and phloem patches, more than 6 xylem and phloem patches, larger and well-developed pith, parenchymatous cortex. Yes, they have more than 6 xylem and phloem patches, or here they can give the condition as polyarch. Option as polyarch vascular bundle. That option can be given because both are one and the same here. So this option can be eliminated because they have more than 6 xylem and phloem patches, which is correct. They have large and well-developed pith. Yes, that is also correct. So this option needs to be eliminated. They have a parenchymatous cortex. Yes, their cortex is made up of parenchyma cells. So this option also needs to be eliminated. 2 to 4 xylem and phloem patches is a characteristic of dicotyledonous root. It's not the characteristic of the vascular bundle of monocotyledonous root. So therefore, option A is the right answer here. So this was about the session wherein we learned about the cells that are present inside the dicot and the monocot root, and also we learned some of the MCQs that can be framed under this particular topic. So I hope you understood the session well. So we shall meet again in the coming session taking up a new topic and discussing the MCQs related to it. So see you in the next session. Thank you.